Good morning. Today we will see the solved question paper of Physiology June 2016 of 2010 scale. So the first essay is describe various events that takes place in the proximal convoluted tubule. What is a normal urine output? So the proximal tubule is the segment of nephron in the kidney which begins from the renal pole of the Bowman's capsule to the beginning of loop of Henle. It can be further classified into proximal convoluted tubule, that is PCT, and the proximal straight tubule. The proximal tubule efficiently regulates the pH of the filtrate by exchanging hydrogen ions in the interstitium for bicarbonate ion in the filtrate. It also responsible for secreting organic acid such as creatine and other bases into the filtrate. Fluid in the filtrate entering in the proximal convoluted tubule is reabsorbed into the peritubular capillaries. This is driven by the sodium transport from the lumen into the blood by the sodium-potassium ATPase in the basolateral membrane of the epithelial cells. 60 to 70% of the filtered sodium load is reabsorbed in the proximal tubule through active transport. Most of ammonia that is excreted in the urine is formed in the proximal tubule via breakdown of glutamine into alpha-ketoglutarate. This takes place in two steps which often generate ammonium conversion into glutamine to glutamate and the conversion of glutamate into alpha-ketoglutarate. The alpha-ketoglutarate generated in this process is then further broken down to form two bicarbonate anions which is pumped out of the basolateral portion of the tubule cell by cold transport with sodium ions. When the filtrate flows through the proximal convoluted tubule, there is an active reabsorption of sodium and chloride followed by obligatory reabsorption of water. So, the osmolarity of the fluid remains the same as in case of Bowman's capsule, that is 300 mOSM per liter. Thus, the proximal convoluted tubule, the fluid is isotonic to the plasma. The normal urinary output is 1 liter per day, 1 to 1.5 liter per day. The second question is what is the actions of thyroid hormone and what is cretinism? The first action is on basal metabolic rate. The thyroxine increases base metabolic activities of almost all body tissue except brain, retina, spleen and the lungs. On protein metabolism, the thyroxine by increasing translation of RNA, by increasing the transcription of DNA to RNA, by increasing the activity of mitochondria, by increasing the activity of cellular enzymes. On carbohydrate metabolism, it increases the absorption of glucose from GI tract, enhances the glucose through the cell membranes, increases the breakdown of glycogen into glucose, accelerates gluconeogenesis. On fat metabolism, thyroxine decreases the fat storage by mobilizing it from adipose tissue and fat deposit. On plasma and liver fat, thyroxine decreases the cholesterol, phospholipids and triglyceride level in the plasma. On vitamin metabolism, thyroxine increases the formation of many enzymes since the vitamins form the essential part of the enzyme. It is believed that vitamins may be utilized during the formation of enzyme. On body temperature, thyroid hormone increases the heat production in the body by accelerating various cellular processes and increase the BMR. On growth, thyroid hormone have general and specific effect on the growth. Lack of thyroxine arrests the growth and increased thyroxine accelerates growth of body, especially in growing children. Effect on body weight. It is essential for maintaining weight of the body. Increase in thyroxine decrease the body weight and fat storage, and decrease in thyroxine cause increase in body weight. Effect on blood. Thyroxine increase the production of RBCs. On cardiovascular system, on the heart rate, it increases the heart rate. On force of contraction, it increases the force of contraction. Blood vessels cause vasodilatation and blood flow increases. On arterial blood pressure, increase the blood pressure as it increases the force of contraction, increase the heart rate. Effect on respiration, thyroxine increases the rate and force of respiration indirectly. On GIT, it increases the appetite and food intake. On CNS, it is very essential for the development and maintenance of normal functioning of the CNS. Cretinism is the hypothyroidism in children characterized by stunted growth. The cause for cretinism are it is due to congenital absence of thyroid gland, genetic deformity or lack of iodine in the diet. Features of cretinism are a newborn baby with thyroid deficiency may appear normal at the time of birth because thyroxine might have been supplied from the mother. But a few weeks after birth, the baby starts developing signs like sluggish movements or croaking sound while crying. Unless treated immediately, the baby will be mentally retarded permanently. Skeletal growth is more affected than soft tissue, so there is a stunned growth with a bloated body. The tongue becomes so big that it hangs down with the dripping of saliva. The big tongue obstructs swallowing and breathing. The tongue produces characteristic guttural breathing that may sometimes choke the baby. Short notes. The first one is structure of cell membrane. Cell membrane is a protective sheath enveloping the body. It is also known as the plasma membrane or plasmalemma. The membrane separates fluid outside the cell called extracellular fluid and the fluid inside the cell called intracellular fluid. The cell membrane is a semi-permeable membrane, so there is a free exchange of certain substance between ESF and ICF. Thickness of the cell membranes varies to 75 to 111 A. Composition of the cell membrane. Cell membrane is composed of three types of substance, that is protein nearly 55%, lipids nearly 40% and carbohydrate nearly 5%. Structure of cell membrane. On the base of the structure cell membrane called a unit membrane, the three-layered membrane. The electron microscopic study reveals that three layers of cell membrane, namely one central electron lucent layer and two electron dense layers. The two electron dense layers are placed on either side of the central layer. The central layer is a lipid layer formed by lipid substance. Other two layers are protein layers formed by protein. Cell membrane contains some carbohydrate molecules also. Functions of cell membrane. First one is protective function. Cell membrane protects cytoplasm and organelles present in the cytoplasm. Selective permeability. Cell membrane acts as a semi-permeable membrane which allows only some substance to pass through it and act as a barrier for other substance. Absorptive function. Nutrients are absorbed into the cell through the cell membrane. Excretory function. Metabolites and other waste products from the cell are excreted out to the cell membrane. Exchange of gases. Oxygen enters the cell from the blood and carbon dioxide leaves the cell and enters the blood through the cell membrane. Maintenance of shape and size of the cell. Cell membrane is responsible for the maintenance of shape and size of the cell. The fourth question is pain pathway. The analgesics pathway with the pain transmission is considered as the descending pain pathway. The ascending pain pathway being afferent fibers that transmit pain sensation to the brain. Gate control theory. According to them, the pain stimulus transmitted by afferent pain fibers are blocked by gate mechanism located at the posterior gray horn of the spinal cord. If the gate is open, the pain is felt. The gate is closed, the pain is suppressed. Mechanism of gate control at spinal. First one, level 1. When the pain stimulus is applied to any part of the body, besides pain receptors, the receptors of other sensations as touch are also stimulated. Level. Then 2. When all these impulses reach the spinal cord to the posterior nerve root, the fibers of touch sensation send collaterals to the neurons of the path, that is, cells of marginal nucleus and substantia gelatinosa. Impulse of touch passing through the collaterals inhibit the glutamate and transmit P from the pain fibers. This closes the gate and pain transmission is blocked. Role of brain in gate control mechanism. According to Melzack and Wall, the brain also plays some important role in gate control theory as the spinal cord. That is, if the gate in the spinal cord are not closed, pain signals reach thalamus through the lateral spinothalamic tract. And these signals are processed in thalamus and sent to the sensory cortex. Perception of pain occurs in cortical level in context of the person's emotional status and previous experience. The person responds to the pain based on the integration of all this information in the brain. Thus, the brain determines the severity and extent of pain. To minimize the severity and extent of pain, brain sends feedback back to the spinal cord to close the gate by releasing pain relievers as opiate peptidase. Now the pain stimulus is blocked and the person feels less pain. Significance of gait control. Thus, gating of pain at the spinal level similar to presynaptic inhibition. It forms the basis for relief of pain through rubbing, massage technique, application of ice pack, acupuncture, and electrical analgesia. All these techniques relieve pain by stimulating release of endogenous pain relievers, that is opioid peptides, which close the gate and block the pain signals. Erythropoiesis, we have seen the answers early. Event at a neuromuscular junction. When the impulse are transmitters from the nerve to the muscle, a series of events takes place in the neuromuscular junction. They are the five events are: first one is release of acetylcholine, action of acetylcholine, development of end plate potential, development of miniature end plate potential, and destruction of acetylcholine. Functions of thalamus. It is a large ovoid mass of grey matter situated bilaterally. Thalamus, primarily concerned with somatic function, is placed with many visceral roles. They are: first one, it is a relay center. Impulse of almost all sensation reach thalamic nuclei, particularly in the ventral posterolateral nucleus. After being processed in the thalamus, the impulses are carried to the cerebral cortex to the thalamocortical fibers. Thus, thalamus is called relay center for sensations. Second, center for integration of sensory impulses. Thalamus form a major center for integration and modification of peripheral sensory impulse before the impulses are projected to the specific areas of cerebral cortex. Then center for sexual sensations, role in arousal and alteration reaction, center for reflex activities, and center for integration of motor functions. Enzymes of pancreatic juice and their functions. The enzymes are proteolytic enzyme, they are trypsin, chymotrypsin, carboxypeptidase, nuclease, elastase, collagenase. Lipolytic enzyme are pancreatic lipase, cholesterol ester hydrolase, phospholipase A, phospholipase B, colipase, bile salt activate pancreatic amylase. Action of trypsin. First one is digestion of protein. Trypsin is the most powerful proteolytic enzyme. It is an endopeptidase and breaks interior bonds of the protein molecules and convert protein to proteose and polypeptides. Curdling of milk, it converts caseinose in the milk to casein, and blood clotting, it accelerates blood clotting, and it activates the other enzymes like chymotrypsin to chymotrypsin, pro-carboxypeptidase to carboxypeptidase, pro-elastase, pro-colipase to colipase. Trypsin also activates collagenase, phospholipase A, and phospholipase B. Autocatalytic action. Once formed, trypsin itself converts trypsinogen into trypsin. Actions of chymotrypsin. First one is digestion of protein. Chymotrypsin also an endopeptidase which converts proteins to polypeptidase. Digestion of milk. Digest caseinogen faster than trypsin. Combination of both enzymes cause rapid digestion of milk. Actions of carboxypeptidase. Carboxypeptidase exopeptidase and break terminal bond of protein molecule. Action of nuclease. Release nucleus, which is responsible for digestion of nucleic acid. The enzyme convert RNA to DNA into mononucleotides. Elastase digests elastic fibers. Collagenase, it digests collagen. Pancreatic lipase, it digests triglyceride into monoglyceride and fatty acid. Cholesterol and ester it convert cholesterol ester into free fatty acid and fatty acid by hydrolysis. Phospholipase, it converts lecithin into lecithin and cephalin to lysocephalin. Phospholipase A, B, it converts phospholipids to phosphoryl, choline and free fatty acid. Colipase, it facilitates digestive action of pancreatic lipase on fat. Bile salt activated lipase, it hydrolyzes a variety of lipids, phospholipids, cholesterol esters and triglyceride. Pancreatic amylase converts starch into dextrin and maltose. Pregnancy test. Pregnancy test is used to detect or confirm pregnancy. The base of pregnancy test is to determine the presence of human gonadotrophic hormone in the urine of the woman suspect of pregnancy. Both biological and immunological tests are available to determine presence of HCG in the urine of the pregnant woman.